are going to see about the different types of centrifugation individually. So, in the case of preparative centrifugation, there is going to be isolation of biological material for the biochemical investigation. So, it is going to be only qualitative, where a different types of samples can be separated off, whether it can be a cell components or a biomolecules. Different types of biomolecules can also be separated in the case of preparative centrifugation. Based on the medium of suspension, that is going to be a single gradient or a different gradient, based on the gradient can be used, these preparative centrifugation can be further have a types of differential centrifugation and density gradient centrifugation. In the preparative centrifugation, the components and the biological molecules are separated, which can be used as a for preparation of any other byproducts further. But in the case of analytical centrifugation, where very small amount of sample can be used for the centrifugation for identification of both qualitative and quantitative level. But in the case of preparative, it can be used for preparation and further techniques. So, a differential centrifugation, where we can found its other name as homogeneous centrifugation, because there is going to be a single gradient to be used. But in the case of density gradient centrifugation, a different types of gradient can be used. Then analytical centrifugation. It is useful for identification of characteristics finding of a biological material. It is helpful in determination of sample purity. Even a small amount of impurities can also be discarded in the analytical centrifugation. Even a small amount of impurity will not be available in the analytical centrifugation. So, a purest components can be separated off, and a qualitative quantitative level can also be possible with the analytical centrifugation. Even a small conformational changes or molecular weight changes can also be separated off in the analytical centrifugation. Due to the a small type of purity, due to the highest amount of purity, a small type of impurity not possible, highest type of purity is possible. It's because of its high RPM. Because among the other centrifugation, analytical ultracentrifugation is going to have a high RPM, so that it will provide more heat. So the vacuum system and